All right, let's move on to part 6. These last two parts are a little bit shorter, so we are almost there. We want to use these last two parts to put the different lectures together. How do these different systems work together? Part 6 will have a cellular focus. Part 7 will have a chemical focus. How can we use this to treat disease? But I do want to show you some of the cutting-edge work that's going on in immunity. So we can understand how TCRs bind MHC. We can identify anti-cancer TCRs just like we can identify anti-cancer antibodies. So can't we give those to T cells and then put them into the person? Part of the problem is you don't want to give one person another person's T cells. You have to harvest the T cells from a person, then put the anti-cancer TCR onto them, and then put them back in. But actually there are companies that are doing that. It's called CAR T cells. Chimeric antigen receptor is what it's called. Because the T cell receptor just needs to be the same on the inside. You can actually mess with the outside of it as much as you want to get something that would be a good anti-cancer binding molecule. And so I've had students who've gone on to companies who have worked on these exact things. And there have been some that have remarkable success. The most success has been against leukemias and lymphomas, the blood-borne sort of immune system cancers. Seems that CAR T-cell therapy worked really well against this at first. And in fact, you look down here, Juno Therapeutics of Seattle was one of the success stories. So we're talking about a local sort of company that was doing this. This is all the way back in 2006, though. As you can see, when you're playing with the immune system, you've got to stay on the balance, right? And if you activate it too much one way, no matter how specific it is, you can have problems. So 18% of the people in a certain trial developed cytokine release syndrome. And you can see that's one of those signaling molecules in the immune system. Their signals went haywire. 2 of the 62 patients died. Now, these were patients that were enrolled because they had no other options. And yet, the same thing is, it's because they knew this was dangerous, and they knew that this field would have a lot of danger to it. And so you can't, it's not easy to change the immune system, and you need to do a lot of trials, and you need to try to figure out how. 14 years after that 2006 event, basically the field built itself back up. They did the safety tests. They figured out one of the things that worked is using a chimeric antigen receptor. That is really an antibody domain. You see how it's a VH and VL linked together antibody domain. So a single chain FV, which means just the 2 first tip domains of the antibody, joined together with a linker and put onto a T cell receptor. Looks totally different on the outside of the cell, but on the inside of the cell, look what it's got. It's got that same zeta chain. And so it's doing the exact same thing that it's showing there. And so you have, and then they experimented with different kinds of insides of the cells to see what resulted in the best kind of activation. And they figured out how to basically build up a new kind of T cell receptor that then you can put into the patient's T cells and turn them into anti-cancer cells. So basically there are whole systems commercially for making CAR T cells. It's really taking off now. It's very intensive and it's very expensive, but it is working much better than I was worried about back in 2006. So there's this is the website you can go to if you want to do your own kind of T cell making. They give you videos about their instruments that they use, and these are really just fancy cell culture and molecular biology type of experiments. And so if you want to learn about this, you can watch the commercials. I just want to say that this is here, and this is all built on the idea of the basics of structural immunology that we've been going through. And let's remember, of course, we've mentioned how T-cell immunity is complex. There's multiple kinds of TH cells. Here's one that just shows helper cells being differentiated into TH1 and TH2 cells. And those have completely different roles. And you see there's a self-antigen role on the top, there's an allergen on the bottom. Basically, I just want to say the same kinds of cells in the middle, but these are playing different roles in different immune responses with different interleukins, different activation patterns of B cells. One of them interacts with macrophages, the other interacts with eosinophils. Basically, there's a whole class of immunology that we can have on that. Good thing that we offer it. And I'm going to be teaching it next year, so I'll be getting ready to talk more about these things there. For right now, just know that these are mediated by molecules. And so if we find the right way to manipulate the molecules, we can actually affect how the disease goes, and we can augment or even make an anti-cancer response in a person. So if we look at these really complex systems, all these types of T cells, all these types of macrophages and dendritic cells, how can we classify them? We've done a lot of experiments where human brains have looked at the data and tried to classify it into groups. With all this data that we've collected, is it possible that computers can look at it? This isn't really AI because it's so dependent on human data. It's not really, I'm sure people are using artificial intelligence to sort through it, but it's really a sorting function. I don't see it as a real intelligent act, but it's more of a correlation act, looking for patterns. And computers can find patterns in complex data. And this is one case where people tried to collect data systematically from 10,000 different tumors. And they tried to see at the level of RNA, they tried to collect RNA, DNA, mutation, clinical data, methylation even. And they tried to see which of all this data went with certain types of cancer and certain types of immune response to that cancer. Could we come up with categories that maybe we haven't seen before? And so it's an incredibly complex paper. I don't recommend presenting it in 5 minutes, but it's that's the basic idea. And the answer is, at least according to the paper, and it got past peer review, so there's something to it, I think, that they found 6 different categories of immune responses that we can sort of group together. And we can say that there's these 6 kinds, 6 colors here of immune responses with different cells, different types of responses to different types of things. This is from the Institute for Systems Biology, which is another local institution. Who knows, maybe they're doing internships someday. This also is related to the Cancer Genome Atlas. This is the idea of let's just collect all the tumors, let's sequence them, let's put all the data together. In fact, let's not just sequence them, let's look at their RNA too. Not just DNA, but RNA and even proteomics, whatever layers we can get to them. So that's the idea of what it was. In 2013, they had 6 different types of omics and 12 tumor types that they looked at. And so that makes 6 times 12, 72 different categories of data they were looking for. They're sifting through that and looking for patterns. And actually you can see the 12 tumor types listed over here if you're interested. And so they worked with this data and they tried to classify it to see if among all these different types of data, were there patterns. And they found 6 different patterns, 6 different colors here. And just notice that they had the ratios between different types of cells. And the characteristics, was there a lot of proliferation? Was there a lot of heterogeneity inside the tumor? And what was the ratio of Th1 to Th2? You see you have these 5 columns, really 4 columns and an other column, but the columns are different for each of the 6 types. And maybe there's some patterns that we can get here. You have an interferon gamma dominant subtype. That way the immune system interacts is different than how it interacts when you have a TGF-beta dominant subtype. And that's different when the red wound healing subtype, which is nice because wounds are red, so you can sort of remember that. Again, a lot of details, but this comes from DNA, RNA, and protein data, looking at what you see in the different tumors and trying to find patterns. They found 6 major patterns, and if you want to someday, you can use this as a start to look at that.